Hello children, I'm so sorry, there was a glitch. Just let me see. Okay. Okay, we'll see if someone is joining over here. And then we'll start off, otherwise we'll stop this. I'm so sorry children. So there was a technical glitch that happened, which is why everything got shut off. Waiting for 2 seconds, otherwise we'll end this. I mean, not 2 seconds, 2 minutes. Okay, I think we should be doing one thing. Yes, hi children, am I visible? I hope I am. Can you tell your friends also that we are using a different link? I'm so sorry for that abrupt ending. Yes, so we were talking about Miller's experiment, right? So in Miller's experiment, what were we doing? Let me see if everyone is here. Okay, let me complete Miller's experiment and then we'll start off. So in Miller's experiment, he was making sure that there was a reducing environment and there was high temperature, 800 degrees Celsius. Okay, so he had a broad sort of thing. From here, he was boiling water, and from boiling water you get water vapor. It was directly sent to a place which was like this. Do not go with my diagram, okay? It can be really bad at some time, or probably all the times. Okay, so basically he had 2 electrodes with the help of which he was generating high temperature, that is 800 degrees Celsius. Okay, and water vapor was here, and also he provided certain gases such as methane, such as ammonia, such as H2 and water vapor. Okay, but make sure there is no oxygen, and this was linked to another thing which went like this, and over here, oh my god, the diagram is really bad, but yes. So basically he found that the inorganic things were given, temperature was maintained high, reducing environment was maintained, and towards the lower end, the organic compounds were seen, such as amino acids and stuff. So where did we get that from? From the inorganic things. So this is chemical evolution, my dear children. Okay, this is chemical evolution. Now going ahead, I'm so sorry for the diagram. Okay, and going ahead with linkage and recombination, first thing. So linkage and recombination, exam point of view, if the amount of recombination is given high, that means linkage is going to be less. Easy thing. Listen, I always tell. So you have 2 best friends. Okay, you are 2 best friends. Now, if you are very close to each other, there is no chance that another person can enter into this particular circle. So I'll say the same thing applies over here. The genes, if they are closely linked to each other, whatever recombination will you do, whatever crossing over you do, my dear children, these guys will be moving on together. So the amount of new combination that will be found will be what? Less. So if your linkage is way too much, that means recombination which is there, it is going to be way too less. On the other hand, I was talking about this, right? Over here, suppose you and your best friends are not actually really close. So the genes are over here. They are distant. They are loosely linked. Now one part of this chromosome can go to another part and create a new recombinant. So when there is loosely packed linkage, when the linkage is less, my dear children, the amount of recombination will be more high. So 50% is the highest, recombination. Now if in the question it is written 37.2, that means here the linkage is way too much. Here the recombination is what? Way too much. If in the question it is given the recombination percentage is 37.2, that means this percentage is way too much. It is loosely linked. The genes are not close to each other, they are distantly located. If it is written 1.7% is a recombination, that means less amount of recombination is there, they are very close to each other. Now other questions that can be coming onto this, okay, on the same field is that who gave down gene mapping? Who said that basically we can find out the location of genes with the help of percentage of recombination all? It was Alfred Stewart. It was Alfred Stewart. He was a student of T.H. Morgan. So T.H. Morgan is a person who found out linkage, okay, and his student Alfred Morgan found out gene mapping. He said if you know about linkage, if you know about the percent of recombination, with that we can find out the gap or the distance between genes as well. Okay, works? Done with that? Okay, I hope Darshan I am able to make you understand that. If it's clear, just give me a heart or thumbs up. Works? And Safura, hi, thank you guys for being here. And bioactive molecules, the next thing. Bioactive molecules are the things being released by certain organisms which are of very high importance. So we have three things in bioactive molecules. One is cyclosporin A. Okay, cyclosporin A is basically released by a fungus, that is Trichoderma polysporum. Okay, this is what? A fungus. What does this do? Why is it helpful for us? It is helpful for us because it is a depressant, or yeah, it is a immunosuppressant, not depressant, sorry, immunosuppressant. It suppresses our immune system. Ma'am, why do we need to suppress our immune system? Basically, suppose I have done an organ transplantation. At this point of time, my body will feel that this guy is foreign, I have to basically remove it out. But I just transplanted it, I cannot remove it out. So at this point of time, I want my body to suppress its immune system a bit. So that is why we give cyclosporin A. Hi Riaz. Okay, Ramesha, I love you too. Okay, now going ahead with the next bioactive molecule, that is statin. Statins. Now statins is produced from Monascus purpureus. Monascus purpureus. Okay. Now what does this statin do? Statin stabilizes the blood cholesterol level. That is how I remember it. Okay, statin which is there, it will be stabilizing or lowers, okay, stabilizing or basically lowers blood cholesterol level. How it basically goes and competes with the enzyme that is responsible for formation of cholesterol. Kalavati, thank you so much. Okay, so these two are there. Apart from that, you have streptokinase. You have streptokinase. Now this is an enzyme, streptokinase. It is produced from a bacteria that is known as Streptococcus. What does it do? What does the streptokinase do? Very important. If you are, if someone, okay, has myocardial infarction or basically heart attack, at this point of time there are high chances that the blood vessels are clotted out. So to break down this clots, my dear children, you need a clot buster, and that is streptokinase. This would be what? A clot buster. Done. So these are the bioactive molecules. These are the bioactive molecules. Ma'am can convert a monkey into Einstein. Oh my god, thank you so much. Okay, explain about pest resistant. Okay, just a second, okay, if I'm missing out anything. No. So I have solved all your doubts. Full panic attack. Pest resistant in okay. Listen. So now I'll talk about pest resistant first, and then we'll go ahead with the next thing. Okay. No need of panic attack. You should not be panicking. I should be panicking. You, my students. What will happen to my students? I should be thinking like that. Well, children, it's okay. Do not, first of all, breathe in, breathe out. Okay. Do whatever you can with the limited amount of time that you have. There's nothing else that can be done. Be happy. You say, I can do it, and you will be able to do it. Okay. So, pest resistant plant in biotechnology is... You're talking about pest resistant plants, or how do you make it pest resistant? Is that so? So one is BT plant. Okay, that is basically what you do. There is a bacteria that is Bacillus thuringiensis, BT. Okay, now this particular bacteria scares off any pest. Why? Because Bt has a gene in it which is known as crygene, and crygene produces what? Crygene is responsible for coding for protoxins, my dear children. So this crygene, my dear children, which codes for protoxin, it has that in its body. Now suppose a pest comes and eats this bacteria. Inside the body of the pest, this bacteria will be going. Inside the body of the pest, this bacteria will be going, and slowly this particular toxin will get activated. Until now it was not, but now it will get activated. Why? Because in the pest midgut, in the midgut of the pest, my dear children, there is an alkaline pH. There is an alkaline pH which is very much important for this to get activated, and it gets activated. And now what happens? It happens in the midgut of the insect. Okay, and slowly the epithelial cells, there are cells surrounding the midgut, right? The epithelial cells are going to be destroyed, and that's how it's going to be died. Now what do you do is that basically, how do we use it? We take the crygenes of the Bt toxin and we insert it into the plant. Now the plant itself will be producing protoxins. You don't need a bacteria to be there. That is one method. Okay, that is one method. Okay. Now what? There is one more method, that is RNA interference. Children, I will be getting back to the MCQ. RNA interference. Okay, what is that? That is done in the case of Meloidigan incognitia and the tobacco plant. So tobacco plant's roots are affected by a worm, that is Meloidigan incognitia, a nematode. Okay, now this Meloidigan incognitia, what it does is that basically it has the mRNA, and this mRNA will be coding for some protein. And this protein becomes highly toxic for the plant, and that's how the plant dies. So basically what do? If the mRNA is not single-stranded anymore, it won't be making protein, because that is a rule. If you're single-stranded, that's when we can do translation and make the proteins. So basically what do? You silence this particular mRNA. You silence this particular mRNA. How? By making it a double-stranded RNA. So what do? You have a double-stranded RNA, you insert this double-stranded RNA, slowly it will break this bond, and this guy, okay, it will form a bond over here, making this mRNA as what? Double-stranded. Now do you think it can undergo translation? No, it does not undergo translation, my dear children. It won't be forming the proteins, it won't be affecting the plants. But how do you basically insert this over here? So what do is that the gene that codes for this particular protein is taken. Okay, it has been taken. It is put in a bacterium that is known as Agrobacterium tumefaciens. Now, Agrobacterium tumefaciens is a, what do you call it? It is a vector. Okay, it is a vector that has the plasmid, and inside its plasmid, we are going to put this particular gene. Okay, now this Agrobacterium tumefaciens is going to affect the plant. By affecting the plant, I mean that it is going to transfer this particular gene of interest into the plant. Now, inside the plant makes its own double-stranded RNA. Plant makes double-stranded RNA with respect to the gene that is being inserted. And this gene belongs to what? This guy, that is the Meloidigan incognitia guy. Now, it makes dsRNA. Every time the worm comes and eats it, basically it is going to take what? dsRNA, double-stranded RNA, and this double-stranded RNA is going to silence the whole thing. Always remember, you need a vector, and the vector is Agrobacterium tumefaciens in the case of plants. In the case of animals, it is disarmed retroviruses. Okay? Ma'am, your name? My name is Jasmine Joy. Okay? My name is Jasmine Joy. Thank you, Srujana. Okay, mutualism with example. Mutualism is basically when both of them get benefit. For example, you're talking about plant association, that is mycorrhiza. In mycorrhiza, there is plant and there is fungus. Plant, photosynthesizes, it produces organic matter. Fungus, what does it do? It gets into the soil, takes in phosphorus and gives it to the plant. So both of them are benefiting. Now, you have fig and wasp. Wasp lays its egg over there, okay, and the fig, it gets benefit because the wasp also does pollination. So like that. Okay. Works, children. Now what? Ma'am, important questions. I had already did important questions in the top 80 plus. Please go watch it. Okay. Shall we move on to the MCQs now? Children, microbes and human welfare. Shall we start? Okay. Let's start. the first question that we have, compare the statement 1 and 2. Children, we have to make it really fast, okay? Thank you, everyone. Okay. Compare the statement 1 and 2. Statement 1 says mycorrhiza is a symbiotic association of algae and a fungi. Mycorrhiza is a symbiotic association of algae and a fungi. Is it correct? Children, tell me the answer now. A, B, C, D. Question number 1. A, B, C, D. Fast. We have to study a lot, right? Okay, no one is telling the answer? No. Yes, the AC is off. It's very hot now. Ma'am, wait, I have a doubt. Yes, ask your doubt. Hannah says 1B is the answer. 1B is the answer. Is it true? Let's see. So statement 1 is false. Is statement 1 false? Let's see. Mycorrhiza is a symbiotic association of algae and a fungi. It's wrong because mycorrhiza is an association of plant and a fungi. Plant roots and the fungi. Now, second statement, they absorb nitrogen from the soil and pass to the plant. Wrong. They absorb phosphorus from the soil. They absorb what? Phosphorus from the soil. So your answer is both the statement 1 and 2 are false. Okay, both the statement 1 and 2 are false. So, both the statement 1 and 2 are false. Until you said this, ma'am, your handwriting is so much good, I realized that everything was true. But after seeing that statement, I realized all of the things that you were saying was absolute lie. Yes, we will be waiting. I will be making a video for that, okay? So it is D answer. Children, please make sure you're getting it correct. Now going ahead with the next question that we have is this. Alexander Fleming discovered penicillin while working on what? Alexander Fleming discovered penicillin while working on what? Staphylococci, which is a virus, Streptococcus, which is a bacteria, E. coli, which is a bacteria, Staphylococci, which is a bacteria. Tell me. What is the answer for this? Alexander Fleming actually was working on Staphylococci, which is a bacteria. Okay, it was a bacteria. And then one day he didn't wash his petri dish. He just kept it like that and he went off. The next day he thought that, oh, it might have formed so many bacteria up there. But he was surprised that it was not happening like that. Why? Because there was a mold being formed. That means a fungus was being formed over there. This fungus was Penicillium notatum. Penicillium notatum. And then he found that probably because of this fungus, the bacteria was not growing. Why? Because it might be releasing something that inhibits the growth. And what is that something, my dear children? It is an antibiotic. What is this antibiotic? This is penicillin. This is what? Penicillin. So he was working on Staphylococci, which is a bacteria. That's when you got it. Okay. Habib Batar, I will be coming to that. Okay, so your answer is D, children. If you are not participating, there is no point of taking this live would say ahead. Okay, I am from, don't spam. Okay, so I am from Indore, Madhya Pradesh, but I am a Malayali. Okay, now question number 3. Which microorganism is useful in production of citric acid? Children, A, B, C, D. Where is Streptococci used? Streptococci is not used. Streptococcus gives streptokinase. I just explained you that, right? Streptokinase is a clot buster. It's okay. It's okay. Now, children, please tell the answers. Which microorganism is useful in the production of citric acid? (3D), yes, Ammu. Good. Vishwa says B. Okay. Hana says C. Okay, Harinakshi says C. Everyone says C. Yes, you are absolutely correct. The answer is C. Aspergillus niger is the one that produces citric acid, my dear children. And this Aspergillus niger is a fungus. Okay, what about Acetobacter aceti? It is producing acetic acid. Penicillium notatum produces penicillin. Clostridium butylicum produces butyric acid. The name suggests everything. This Aspergillus niger is something that is different, that produces citric acid. Great going, children. Shweta, Kalavati, Ashish, Vishwa, Lakshmi, Hana, everyone. Okay, great going, great going. Shripad also. Okay, now going to the next question that we have. Activated sludge is the sediment deposited in. Activated sludge is what? The sediment deposited in what? Now there was a question of someone asked, Habib Tar, microbes in sewage treatment. So I'll be explaining the sewage treatment, just go through it once, okay? And others, please answer what is the fourth answer. Fourth answer, what is it? You can tell me. So listen, first thing, in treatment you have primary treatment. You have what? Primary treatment. What do you do in primary treatment? You do two things. One is filtration, and then the next thing you do is sedimentation. Filtration means whatever things can be removed out which floats will be removed out through filtration. Sedimentation means the one that will sediment down will be removed out like that. Whatever you're left with is known as primary effluent. What? Primary effluent. This primary effluent is put in an aerobic tank. Is put in an aeration tank. Okay, not aerobic tank. It's put in an aeration tank. Aeration means there will be too much amount of air in there, and in this air, my dear children, who will survive? It's the aerobic bacteria that will be surviving. So aerobic bacteria is not going to survive and going to act on the primary effluents, organic matter, the organic matter present in the primary effluent. When it does so, it needs a lot of oxygen, and this is what you mean by biochemical oxygen demand. When there is organic matter, that's when aerobic bacteria will need too much amount of oxygen. That means BOD is way too much. So if BOD is way too much, that means the water is polluted. Now, if the organic matter is not too much, that means aerobic bacteria will need less amount of oxygen. That means BOD will reduce. So this happens until the level of BOD gets reduced in the aeration tank itself. And slowly the aerobic bacteria is going to be friends with fungal filaments and lot of other things, forming something that is known as flux. It forms flux. Now what do is that you put it in a settling tank. This is your answer. You put it in a settling tank, my dear children. In the settling tank, what do is that, okay, what do is that, basically this flux and everything will be settling down, okay, and whatever treated water is there, it will be over here. You can put it down to the river or the stream or whatever is there. What do you call this as? This is known as activated sludge now. You call this as activated sludge. A little amount of sludge, you take it back and put it in the aeration tank because you want to make more aerobic bacteria for the next batch that is coming. That is known as inoculum. It acts as an inoculum. The rest of the things, what do? So you have removed the water and everything to the river and the stream and everything. Now you put this activated sludge in an anaerobic sludge digester. Anaerobic sludge digester. That means there is no air up here. There are anaerobes, such as methanogens, and they will be acting on the aerobic bacteria and everything, and will be releasing what? Methane. Will be releasing methane, carbon dioxide, H2S and everything together, forming the biogas, and from the activated sludge you're getting biogas as well. This is known as what? The next treatment which is there. We can't hear your voice. Can you just check? Others, can you hear my voice? So your answer is activated sludge is a sediment deposited in what? Settling tank, and with that I explained the whole thing. Okay. Okay. Children, is my voice actually breaking for everyone? Is my voice breaking for everyone? Can you tell me in the session? Okay. Yes. No. Okay. Rishwa is saying no. So others who are saying yes, can you just recheck your wiring and all things like that? I think it will be working. Okay. Now match column 1 with column 2 and select the correct option using the codes given below. Citric acid is produced by what? I just told you right now. Thank you, Pankaja. Okay. Tell me, children. Children, just let's be restricted to studying as of now. Tomorrow you have exam. Okay, I'll put the mic over here and check if it's working. Fine. I can hear you clearly. So children, it's your ear, what do you call it? The earphones problem. Please check it once. So Shripad says 5B. Everyone says C. Okay. Manjula say, okay. We are not able to hear. Can you please check? Okay, Akshu is now able to hear, I suppose. Shripad says B, C, everyone. Okay. Listen, citric acid is produced by Aspergillus. Cyclosporin A is produced by Trichoderma. Statins is produced by what do you call it, Monascus purpureus, butyric acid by Clostridium butyricum. So A goes with 3. A goes with 3, you have 3 options like that, so exclude this one. Okay, now B goes with 1. B goes with 1, you have 2 options like that, so exclude this one. Eliminate this one. Okay, now statins is produced from Monascus. C goes with 4, this becomes your answer. B is your answer for this. Okay. Okay. NHKOP is live YT in YouTube. Right. So your answer is B. I hope you got it correct, my dear children. All of the children who are watching this. Yes. Let's move ahead. Now, during sewage treatment, the gas produced in anaerobic sludge digester includes what? Anaerobic sludge digester includes what? Hydrogen sulfide, methane, or what? This is 6th question. Children, direct questions are there. So let's make it fast. The gas produced in the anaerobic sludge digester, as in where there is no oxygen, what are the gases that is being produced? All of it comes together to form the biogas. All of it comes together to form the biogas. Vishwa Rathod, I hope you're saying for 6 only. It's C. Shripad says it's B. Arali says it's C. Riya says it's C. Sharad B. Gauri C. Lot of answers, different answers. My dear children, it is methane, hydrogen sulfide and carbon dioxide. This is your answer. All of this together becomes the biogas. Please, if at all you're going and writing that the biogas is being made by just methane, you're going to get yourself wrong. So all of these gases comes together, my dear children. Methane, hydrogen sulfide and carbon dioxide, it forms the biogas. See. Cool? Going ahead with the next thing. Which of the following is not an advantage provided by a mycorrhiza to the plants? Is not an advantage. Okay, Kalavati, great. Is not an advantage provided by mycorrhiza to the plants. Mycorrhiza is a combination of plant and fungus, right? So, apart from providing phosphorus, it provides resistance to root-borne pathogen, that is there. It helps in tolerance to salinity and drought. That means if too much amount of salt or less water content is there, it provides tolerance to that. Now, overall increase in plant growth and development, that is also being provided. Okay, that is also being provided. But it does not have anything to do with increased in yield quality. So, B is the answer for this. 7, answer is B. In your NCERT, there are lines being mentioned and you might not, take a hold of it, but this is the answer. Okay, 7 is B. It does not have anything to do with the yield quality. Remember one more thing, when you talk about biogas, what were the groups which gave down the biogas plant and everything? It's there in the NCERT, KVIC, something like that, Indian Agricultural Research, something. So just go through it also, IARI, very important thing. Okay, so maybe everyone says B, it's so good. Next, so the microbial biocontrol agent for butterfly caterpillar is butterfly caterpillar. What is a biocontrol agent for that? What kills the butterfly caterpillar? It's an organism only. You do not need pesticides. Okay, you're not using pesticides. Instead, you're using this. So, what is the answer for this? 8th. Bacillus thuringiensis, Saccharomyces, Cyanobacteria, Staphylococci. What is the answer for this? The answer for this is Sia. Lata, Shripad, Vishwa, okay, Akshu. Okay, children who are saying Bacillus thuringiensis is the answer, you get it correct. I told you Bacillus thuringiensis BT. Okay, it has cry gene, and the cry gene are strictly focused on what? Killing insect caterpillars and larvae and things like that. Okay, you have different types of cry genes. You have studied this in biotechnology principles and processes, or biotechnology and its application, either way. Okay, so your answer is A, not D, not anything else. A. Okay. Remember all the biocontrol agents. For aphids, what are used? Ladybird is used. For killing mosquito larvae, you have dragonfly. Okay. For killing other insects, you have baculoviruses. That is from nucleopolyhedroviruses genus. You should know all that. Okay, Sharad, thank you so much. Now, which of the following statement regarding phlox is incorrect? Phlox is what I told you, a combination of what? Aerobic bacteria and fungal filaments and certain extracellular things. So, they are the mass of bacteria associated with fungal filaments. So, this is correct. Produced in the aeration tank, yes. Okay, they consume major part of organic matter, yes. Found by useful anaerobic microbes, no. Is by aerobic. So, your incorrect statement is this. Your incorrect statement becomes this. Okay, children, please, as I said, Amu is saying about efforts and all, yeah, please go through that biocontrol thing. It's very, very important. Microbes in human welfare, if you read the chapter really well, no, you get straight 7 marks. So there is no way to twist the answers and questions and give you. Very nice thing. Okay, going ahead to the next question that we have is, select the correct statement from the following. Biogas is produced by activity of aerobic bacteria on animal waste. By anaerobic bacteria, my dear children. Methanobacterium is an aerobic bacterium found in the rumen of cattle. First thing, methanobacterium is an anaerobic bacterium. Anaerobic bacteria. Okay, it's not aerobic, so this becomes wrong. Is methanobacterium found in the rumen of the cattle? Yes. It is an anaerobic bacterium, but it is found in rumen of the cattle, my dear children. You should know that. And what does it do? It helps in the breakdown of cellulose. Cellulose is a part of plant cell, and to break it down, you need methanobacterium. Indirectly, it helps in the breakdown of cellulose. Now, biogas, commonly called gobar gas, is pure methane. No, it is methane plus H2S plus carbon dioxide. So this is also wrong. Activated sludge in settlement tank of STPs is a rich source of aerobic bacteria. Is it? Activated sludge? Thank you so much. I hope it becomes easy than chemistry. I thought a lot of you had a complaint that chemistry was difficult. Lunch, ma'am, I didn't have yet. So, activated sludge in settlement is a rich source of aerobic bacteria. This is correct, my dear children. That is why you use it as inoculum, right? So, thank you so much. NHK OP is live. Okay. Chalo. Bioactive molecule cyclosporin A is produced by what? Yes, take a 5-minute break and drink water. Okay. Bioactive molecule, that is cyclosporin A, is produced by what? Fungus, bacteria, virus, protozoa. Vishwa, what are you saying? How? I didn't get you, please. Can you explain a bit? (11A) But is a fungus. Yes, exactly. Let's see 11A or not. So, bioactive molecule cyclosporin A is produced by Trichoderma polysporum, which is a fungus. And what does it do? It is an immunosuppressant. Cyclosporin A is immunosuppressant. Awesome, my dear children. Great, great going. Okay. Now, going ahead, which of the following, okay, this is a model paper question. Which of the following water samples in the table given below will have a major concentration of organic matter? So, A is this. So, level of pollution is high, the value of BOD is high, low, whatever is it. Tell me. I told you, whenever you have too much amount of organic matter, to break this organic matter, you need what? Too much amount of oxygen as well. You need too much amount of oxygen as well, O2 as well. You get it? So, basically that means biochemical oxygen demand is also high. That is, requirement for oxygen is basically biochemical oxygen demand. So, pollution, organic matter is high, that means pollution is high. So, if pollution is high, my dear children, how do you recognize it? By high level of BOD. What is the answer for this? Sharad, bye. Vishwa, you want 10th question once again. This is the 10th, no? Or did I make it wrong? I'll explain, okay? I'll get to you. I'll get back to you. Tell me the answer for this first. So, your answer is A, okay? Your answer is A. Sharad, bye-bye, take care. I hope you got it Y also. Okay, now, okay, I'm so sorry, there was 10 question again. Okay, so you want me to explain this, Vishwa. So listen, biogas is produced by the activity of aerobic bacteria on animal waste. This is wrong because it is produced by anaerobic bacteria, not aerobic bacteria. Now, methanobacterium is an aerobic bacteria? No, it is an anaerobic bacteria again. And actually, methanobacteria is an anaerobic bacteria which is found in the rumen of the cattle. That is correct, but this part is wrong. Now, biogas, commonly called gobar gas, is pure methane. This is wrong, okay? C is not the correct answer. Biogas, commonly called gobar gas, is pure methane. No, that is also wrong because it is made up of methane, H2S and carbon dioxide. And activated sludge in settlemental tank of STPs is a rich source of aerobic bacteria. That is correct. Activated sludge Okay, the one that settles down, that is flocks and all. It is a rich source of aerobic bacteria. That is correct. So this is the only correct option. D is the correct one. Okay. Okay. Now, if a patient has undergone myocardial, my eyes are little paining. Oh, so you should be stopping the video or watch it later. Okay. Please go. So, if a patient has undergone myocardial infarction leading to heart attack, to remove clots from the blood vessels of the patient, which enzyme is used by the doctor to treat the patient? I told you, clot buster. The clot buster, what is the answer for this? The clot buster enzyme. A. Is it A? Yeah. It's not A. B, exactly. It is what? Streptokinase. Streptokinase, you get it from Streptococcus, which is a bacteria, and that is why it is going to act like that. NHKOP is it's a wrong answer, Baba. It's not C, it's B. Okay. And with that, we complete that chapter also. My dear children, biotechnology principle and processes. With that, we will be stopping in the live. Okay. Let's get ahead. The first question: which of the following is not a feature of plasmid? Which of the following is not the feature of the plasmid? Extrachromosomal, circular structure, single-stranded, independent replication. It's not the feature of the plasmid. Plasmid is what? For example, this is an E. coli. Children, if you're not participating, it won't be easy for me to tell the answer. Please participate, okay? If this is an E. coli, it will have its own chromosomal DNA which codes for everything. Now, it will also have an extra chromosomal stuff as well. That does not code for any protein, it's just there. It has its own origin of replication, it divides by its own. You get it? So, basically, extra chromosomal is correct, circular structure is correct, independent replication is correct because it has its own origin of replication. But single-stranded is wrong. So, your answer is C, not D. Ramesh, I love you too. Okay, so it's D, sorry, it's C, not D. C, 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 C. Yes, great. Now I see the what's it, vigor. You should be participating. Then only when I can have the energy, right? Next thing, the procedure through which a piece of DNA is introduced into a host bacterium is known as. A piece of DNA you're inserting into the host bacterium, into another organism. Basically, don't you think this will be now transformed? Since it has our thing, so it is now transformed. It's a new thing that is being formed. So this is known as transformation. Again, it is what? C. 2, 2, 2. Second question, what is the answer for this? Second, it will be C. Cloning means you're basically making multiple copies. Amplification means again you're increasing the number. Restriction has no point over here. So your answer is C. Okay. Going ahead to the next question. Children, I'll be a little fast now. Okay. The commonly used vector for cloning genes in animals is. The commonly used vector for cloning genes in animals is what? In animals. In animals, you have one very famous vector. What is that? The answer for third question. Kalavati, Shishwini, Ashwini, Meghna, everyone, Ansi, Vishwa, Naveen, great going. C, yeah. B, C, tell me the answer. What is the answer? It is, listen, whenever you want to transfer something to the animals, and if you are using vector, then it has to be disarmed retrovirus. It cannot be anything else, but it has to be disarmed retrovirus. If it is a plant, then you use Agrobacterium tumefaciens. If it is a plant, you use Agrobacterium tumefaciens. If it is animal, you use disarmed retrovirus. So this becomes your answer. Okay, so the answer is B. Yes, TI plasmid. So I'll tell you how does it work in the case of plants. Agrobacterium tumefaciens is a bacteria that affects the plant actually. How does it affect? It has a plasmid which is known as TI plasmid, tumor inducing plasmid. As the name suggests, it induces a tumor. How? Basically, it has a segment which is known as tDNA, transfer DNA, tDNA, okay, tDNA. This segment is basically when this organism affects the plant, it is going to transfer tDNA into it, and this tDNA is going to cause tumor in the case of plants. But now what have we done? We have removed the tDNA part. Instead, we put whatever gene we required. Similarly, it will go and infect, does not know that it does not have the gene to infect. Okay? And that's how it works. Ammu Nayaka, I have said your name now. Okay? So this is disarmed retrovirus. Please remember that. Going ahead to the next question that we have. Restriction site of EcoR1 enzyme is what? EcoR1 enzyme, restriction site of EcoR1 enzyme is what? Fourth question. A. Ashwini says A, Vishwa says A. Is it A? Restriction site as in, you know, any enzyme will not randomly go and cut any place. It will have a particular site that is known as restriction site. And for EcoR1, the restriction site is 5-GAA-TTC. It is 5-GAA-TTC. GA-TC. Okay? Remember that. Cool? Now, if you're asked about HindII, you know, HindII was the first restriction enzyme to be found out. What is the restriction site for it? There is no restriction site that is not mentioned in your NCERT. It has 6 base pairs. It has 6 base pairs, please remember like that. Here also you have 6 base pairs, 1, 2, 3, 4, 5, 6. Similarly, HindII also has a recognition site of 6 base pair. Okay? Next question. The extraction of separated bands of DNA by cutting out them from the agarose gel. You know, in gel electrophoresis, they run through the agarose gel, right? Now, if you want to remove this DNA out of it, what do you call this process as? When you want to remove this thing, what do you call it? The extraction of separated bands? Children, A, B, C, D. C. Hana says C. Yes, it is C. It is known as elution. It is known as elution. Okay, please remember, very important thing. Ammu, Shripad, okay. Shripad, it's not D, Baba, it's C when you're extracting the separated DNA. Okay, going ahead. The thermostable enzyme DNA polymerase is obtained from. Thermostable as in there is a DNA polymerase enzyme version which is able to survive at high temperature also. Okay, where do you get this from? And this is used. Okay, this is used in PCR. This is used in PCR. So what is this? Mithun, okay, sorry. Sixth answer, okay, 6C, okay, 6C, Yashashwini, Hana. Hana says it's B, Ashwini says it's B. Let's see. Yes, Adit says it's B, Naveen Kumar A. Children, it's B. Thermus aquaticus. Thermus aquaticus is the bacteria from which you get DNA polymerase, which is highly thermostable. It is also known as Taq polymerase. It is known as Taq polymerase. Okay? Got it? Children, I'm not able to read out the who all said the correct answer. I'll be reading it out. I'll give you a question. This is the question. Solve it. Agrobacterium tumefaciens transform normal cells into a tumor by delivering a piece of DNA known as what? What piece of DNA does it deliver? And until then, I'll tell you the options. So I have 6B. Ashwini said the correct answer. Vishwa also said the correct answer. Kalavati said correct. Hana, Ammu, Vishwa, Adit, call me something, okay, Mithun, great, all of you gave the correct answer. Okay, now, Srinivas, Ammu, everyone says C, Kalavati, I told your name, da. Okay. So it will be C, my dear children. Agrobacterium tumefaciens transform normal cells to tumor by actually delivering what? tDNA. Transfer DNA. And it's a part of tumor inducing plasmid. Okay, so your answer is C. Again, I'll be reading out your name once I go ahead with the next thing. Identify the correct statement which is put down over here. HindII, you have to find the correct, underline the correct one and then only go ahead. HindII cut the DNA at specific sequence of 6 base pairs. Microinjection is a technique of introduction of rDNA into the plant cell. Biolistics is a technique of introduction of rDNA into animal cell. Retroviruses are commonly used cloning vectors in plants. You have to find what is correct. Tell 8 A, B, C, D. If you're not telling with the digit, I won't be able to understand it. Please, if you have already written the answer, give it once more. Give it once more, but please put it with numbers like 8, A, B, C. HIV is important, cancer is important. Hana says 8A, Srinivas says it's 8A. You have to find the correct statement, okay? Okay, Ashwini says it's B, Hana says A, Ashwini says B. Oh, okay, Kalavati says it's A, Call Me says A, Ying Yang says A, okay, Adit says A, Mithun says A, Ammu says C, Srujana says A. Okay, children, answer is A. Hint to cut DNA at specific sequence of 6 base pairs. This is the only correct statement. Why? Microinjection is a technique of introduction of rDNA into the plant cell. Wrong. Microinjection means you're going to inject something into the nucleus directly, and that is done as a vectorless method for animal cells. For animal cells, when you have to transfer something to the animal cell, you use microinjection because it does not have the cell wall. For in the case of plants, you use biolistics or gene gun method because it has to pass through the cell wall and get into the nucleus. And in biolistic, what do? You have gold or tungsten, small microparticles. You cover it with the DNA and you charge it with high velocity. It gets inside the nucleus. So this is not done in animal cell, this is done in plant cell. Both of them are vectorless methods, one for plant, one for animal. Always remember, biologistic, that is gene gun for plant, and the other one for animal. There is electroporation done for bacteria. Now retroviruses commonly use vectors in plants. Wrong, I just told you it's for animals. It's for animals. Retroviruses are for animals. So this is again wrong. This is the only thing that is correct. Okay, so very much important, vectorless methods are very much important to learn. Okay. Now, which of the following is a stain used to visualize DNA fragments in gel electrophoresis? In gel electrophoresis, it's okay. Please be okay. Which of the following is a strain used to visualize DNA fragments in gel electrophoresis? Cotton blue, ethidium bromide, acetocarmine, safranin. It is, yes, for EcoR1 and not EcoRI, sorry. Escherichia coli, not EcoR1, okay? Hana says it's B, Ashwini says it's B, Adit says it's B, Mithun B, Call Me says B, Srinivas B, Vishwa says B, Hana again, okay. Adit, Manya, okay, all of you are saying B, Sinchana also B. Children, yes. It is what? Ethidium bromide, which is there to visualize the DNA fragments in gel electrophoresis chamber. And also please mention it is put under UV light, then only you can see it, otherwise you cannot see it. And when you put it under UV light, it comes in what? Basically, it gives a bright orangish color. Okay? It gives a bright orangish color. Now, going ahead. Which in the given diagram of PBR 322 vector identify the restriction sites A, B, C and D. What is the restriction sites A, B, C and D? So if you know PBR 322, you will know the answer as well. So over here, listen. This is PBR 322. Okay. Oh, just a second. Low on energy. Jasmine ma'am has become low on energy. Bright orange color. Okay. When under UV radiation, it will give a bright orange color. Now listen, you have to make 2 blocks like this over here. This is the origin of replication, my dear children, of this particular plasmid. This is the ROP site. Basically, it's a protein that is required for, for DNA polymerase, DNA replication to happen. Now over here and here, you have 2 antibiotic resistant gene site. One is ampicillin resistant gene site and the other one is tetracycline resistant gene site. Okay, now over here, my dear children, you have the recognition site for certain enzymes also. So when you have these guys, that means basically they will be resistant to the antibiotic ampicillin and tetracycline. They can survive in both this medium. So these are selectable markers. Now over here in the same area, you have the sites for the enzyme cutting also. What all enzyme? It is BAM H1 site. This is BAM H1 site. This is SAL1 site. Okay, this is SAL1 site. Over here also you have 2. One is PVU1 and the other one is PST1. Now there is PVU2 also, which is there in the ROP site. PVU2. Now here you have 3 enzymes. One is EcoR1, one is CLAV1 and the other one, which looks like this, is basically HIND3. HIND3. Okay, now over here you have to find A, B and C. A, B and C. So A is what? ORI site. A is what? ORI site. So A is ORI site, there is only B option with that. I'm so sorry. So there is only B option with that. So A is ORI site. What about ampicillin resistant? Ampicillin resistant site is over here. So B is ampicillin resistant. Okay. What about C? C is tetracycline resistant, and D is what? HindIII. D is what? HindIII. Here you have 3 enzymes. One is mentioned over here, that is HindIII. So your answer is this. Okay. Now, going ahead. Statement, plasmid DNA acts as a vector to transfer the piece of DNA attached to it. Plasmid DNA which is there, okay, it is acting as a vector to basically attach whatever, to basically transfer whatever is attached to it. Is it true or not? It is true. It is true. Prajwal, I just made it, just go through it once, okay? It is true. Now, vector DNA acts as plasmid to transfer the piece of DNA attached to it. Vector DNA, that means vector's DNA acts as plasmid to transfer the piece of DNA attached to it? No. The vector can be anything. It does not necessarily have to be a plasmid anytime. So this statement is false. Only one statement is correct, that is statement 1. Okay, that is statement 1. So statement 1 is true and the statement 2 is false. So your answer is A. Your answer is A. Geeta Togari and Yashashwini, you are correct. Adit, okay, every one of you are correct. Okay, the steps of recombinant DNA technology are given below. You should be knowing the steps, okay. So first thing that you have in recombinant DNA technology is what? For example, I have a cell. Inside the cell there is RNA, there is protein, there are a lot of things. You have to separate RNA, proteins and everything, okay. So, and you get the DNA, that is isolation of DNA should be the first thing. Okay, you remove the RNA by RNase, you remove the protein by protease, you remove the cell wall first. Okay, in plants you have different enzyme, that is cellulase, in fungi you have chitinase, in bacteria you have lysozyme. Please remember all these names. Okay, enzymes names are really important. Please explain 11th question once more. 11th question once more. This one. So plasmid DNA, suppose this is a bacteria. Over here there is a plasmid DNA. If I am attaching something to it, obviously this plasmid DNA is going to act like a vector. It is going to transport the piece of DNA that I have just attached to it. Now vector DNA acts as plasmid. So example, I am taking a vector. Okay, suppose this is the vector again. This has an E. coli. E. coli, wait. Okay. This is an E. coli which just has its own chromosomal DNA. Can this DNA act like a plasmid? Can this DNA act like a plasmid? No, my dear children, it cannot. So which is why statement 2 is false, statement 1 will be correct. Okay. Okay, I told you the answer also. So basically over here isolation of DNA is the first step. Okay, the second thing that you have, this is the first thing, okay. Second thing that you have is, you are going to cut the DNA at specific locations. You took the DNA, you're going to cut it at specific location, and then you are going to make it run through the electrophoresis chamber, gel electrophoresis chamber. Okay, you're going to run it through gel electrophoresis chamber. Once you have separated that, okay, once you have separated the wanted thing, the smaller fragments, you're going to amplify it. You're going to amplify using a PCR machine. Once you have amplified it, what do? You're going to insert whatever is needed into the recombinant host which is there. And after that you will get your product, and the product will be isolated from every other thing, that is purification and separation will be done in downstream processing. So your answer becomes what? 12D over here. Okay. Ammu Nayaka, can you please go and just check it out once? Okay, there is zero energy left for me to explain anything now. Okay. Now, a piece of alien DNA cannot multiply itself in the progeny cells of the organism due to what? Cannot. A piece of alien DNA, if I just put it there, it cannot. Do you think it will still multiply? It would not multiply that because it does not have an ORI site, origin of replication. And if origin of replication is not there, there is no point of what? There is no chance of replication to get started. They cannot multiply. So your answer for 13 becomes what? A. Your answer for 13 becomes what? A. Okay. 13 is not D, 13 is A. That's the use of origin of replication. And I think this is the last one. Yeah, use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as what? So for example, American company or US company was basically saying that basmati rice is theirs and they were selling it and they were going to take the patency for it. But they didn't give compensation to any Indian farmers who actually bought it. So get it? This is what biopiracy. This is known as biopiracy, my dear children. So 14th answer is A. Similarly, you have bioprospecting in some other chapter. Just go through it once. And with that, we complete that also. And I am going to stop it over here. You have certain questions, okay? You have certain questions from biotechnology application. Now all the other 4 chapters which are left, reproductive health, okay, and organism and population, biodiversity and evolution, biodiversity and conservation, and ecosystem. So I'll request you to go through the PDF. I have already explained all the 1 marker in the all 1 marker series. You can just target these chapters and just go through it. Okay, I'm not going any further because my energy is down, low down, and I won't be able to, go ahead, I think. Yes, so the same questions are there, but please make sure you're practicing model question paper things. So just to sum up everything, I want to tell you one thing. Please make sure you're going through model paper 1, 2 and 3. Very, very, very important. If you have not got time to watch all the big videos that I have made, at least download the PDF and go through all the answers one. But listening to the video will be really good. In that way, you might remember better. Either way, you just see. Okay, practice everything. You are going to go really, really good. I will pray for all of you. Okay, so please do not be worried. And if you like the video, do not forget to like, share and subscribe the channel as well and recommend it to your friends. And also, if you have any doubts, put it down in the comment section. I'll be solving it. Yes, so that's all. Guess I'll have food. Thank you so much, children. Okay then, bye-bye. Ammu, Ashwini, Hana, Vishwa, continuous, I didn't get you. Naveen, thank you so much. Adit, okay then, bye. Kalavati, all, everyone, okay, bye-bye, take care.